but it's another thing. Nothing really matters behind the shades of blue.
Hey guys, welcome back. Thanks for tuning in again. Hopefully the last video wasn't too dry. If you're still here, then I guess it wasn't too bad. As promised, I'm going to be covering biochemistry in this video, and I want to show you really quickly the main source of information that I used when I was double checking all of my work. Basically, it's a book I used when I was in college, Principles of Biochemistry, 4th edition. Actually, it's a really good source of information for the topic of biochemistry. It has really good visuals. It has all the detail you could ask for. So anybody who feels like double checking my work or whatever, or if they just want to learn more, I would really recommend that's a good reference book for that topic. I'm going to be covering 3 major cell functions for this video, and that's going to be energy production, which is how a cell turns sugar in the form of glucose into usable energy by basically converting glucose into ATP, and I'll walk you through that pathway. And then I'm going to be covering protein production, which is really the most important part of this video, and how we go from your DNA, how it's wound up in the form of chromosomes and stored that way, all the way to the point to where the information is translated into proteins. And lastly, cell division. And I'm not going into that topic too deeply, but I want you guys to see the most important part on that topic is just how your DNA from one cell from the parental cell to the offspring, how it's just a one-to-one copy, and physically how that process takes place. Why am I covering these 3 topics? Because there's so many different things I could cover with biochemistry. But I'll just explain here why I think these are good ones to look at. For the topic of energy production, going from sugar to usable energy, I'm covering that specific pathway because it's a very fundamental biochemical pathway. You find it in all cells, and it's a pretty universal pathway. And I want you guys to understand really what a biochemical pathway is. Try not to be intimidated by it or anything like that. Just understand the way. Basically, it's a chain of chemical reactions that takes place to perform a certain task. And like I said, this is a really basic, really fundamental one. And I want to show you how even the simplest task inside of a cell is it's still really complicated. It's still it's a highly regulated series of chemical reactions. There's nothing magic that happens inside of a cell. It's just, it's all chemistry, and we understand the chemistry at this point very well. So I want to show you how we know these things and introduce you to the concept of a biochemical pathway. As for protein production, like I said, that's really the big topic for this video. And I'll try and give you guys as many visuals as I can. This stuff's easier to understand when you can actually see it. But I wanted to show you how very physically, literally physically, DNA is the programming of the cell. And I want to show you how, starting with your chromosomes, which is a way of storing DNA inside of the cell, that DNA is unpacked and then read and then translated, and that literally programs it's basically the instructions for protein production inside the cell, and it can do some other things, but that's the main job of DNA. It's the programming for protein production, and that's really important because with the topic of evolution, everybody's interested in the idea of genetic mutations, which is how DNA changes and how that could come up with new traits. And I want to show you the physical nature of that and what would have to take place in order for a genetic mutation to actually create like a new protein or something like that, and show you how pretty much how ludicrous it really is to think that could lead to brand new traits inside of a cell. But I'll get into that. And lastly, I'm covering cell division lightly because I basically just want to show you how DNA is passed one-to-one from the parental cell into the offspring. When a cell splits off, you can see how the DNA, the chromosomes, are copied, and then they're lined up in the middle of the cell and they're pulled apart, and you can literally trace the information being passed from the parent to the offspring, and just give you some visual so you can kind of see how that works. That's kind of my goal with that. So what do I really want you guys to get out of this video? Because there's a lot of information I could throw at you, and I will be giving you quite a bit, some of it that you've probably never seen before, and I want to try and put that into a context so you know what the purpose is and how that applies to the theory of evolution. First and foremost, what I want you to understand by the end of this video is that things don't just happen inside of a cell. Cells do not have willpower. Cells do not have decision-making ability. Cells don't make choices. And when you take that from the cellular level up to an organism, a full animal or plant or whatever have you, the same thing still applies. They can't just decide to adapt or evolve or mutate to better suit their environment. That's just physically, that's not how it works. Second off, I want you to understand that everything that happens inside of a cell is a tightly regulated, highly controlled chemical reaction. You can boil all of biology in basically all study of life down to a network of chemical reactions, and that's really what biochemistry is the study of these chemical reactions that drive life itself. There's, like I just said, it's all just chemistry. There's no magic involved. And I want you to see how interwoven and how complex these chemical reactions are, these biochemical pathways. And you can kind of think of a cell like a computer. It has a lot of different moving parts, and they all have to work together at the same time, doing their job correctly, or else the whole thing fails. And maybe I'll expand on that a little bit more as we go along here. And the last thing I want you to get out of this is I want you to understand, specifically with DNA, that's really the big focus here, how DNA is literally the information source of the cell, and it's basically responsible for everything that happens inside of a cell. So you have to understand the process of how DNA goes from being wound up in a chromosome all the way to the final product, which is protein production. And in order to understand evolutionary theories and really think for yourself about whether you think that's possible, you have to understand the process of genetics and how DNA programs the cell to make different proteins and regulates those proteins and things like that. So those are the three things I want you to get, that things don't just happen inside of a cell. There's chemistry behind it. That chemistry is very tightly regulated and interdependent, and that DNA is responsible for all information inside of the cell, all protein production, and things of that nature.
Nothing's gonna make you stop, and nothing's gonna break you off. Everybody knows you're lost in the nighttime hunt. We are dead until...
Let's go ahead and get into the first topic here, which, as I said, is energy production inside the cell. So what I'm going to do is take a quick look at how the cell takes glucose, which is a common form of sugar, and chemically modifies it into a more useful form of energy. The major energy molecule inside of the cell is known as ATP, which is adenine triphosphate, and that meets most of the cell's energy needs. It's very useful. It's very accessible energy for the cell. And cells can generate ATP by turning glucose into ATP through a series of different biochemical pathways. And I'm going to do here is cover the pathways, basically start to finish, how you get from one glucose molecule into quite a few ATP molecules that the cell can use to do work later. The first step is a pathway called glycolysis. And what glycolysis does is takes this glucose molecule and turns it into pyruvate. You also get byproducts of NADH and ATP. I'll get more to NADH in a minute here. So let's go ahead and take a quick look at the chemical structure of glucose. It's pretty straightforward. It is a 6-carbon ring, and it has a couple of groups off to the side there, alcohol groups. And this does have some useful energy to it, but not in the way that the cell's machinery allows it. So it has to be broken down into a different form of chemical energy. And in order to do that, the first step, like I said, is glycolysis, where it's a 10-step pathway where this ring molecule is broken down into pyruvate, NADH, and ATP. Each of these steps is modified by a different enzyme. And what an enzyme is it's a protein that performs a certain task, basically. It's a protein with a specific shape and a specific chemistry to it that allows it to passively, just as a chemical reaction, allows it to modify other proteins or substrates or other things like that. And each of these 10 steps requires a different enzyme. And start to finish, you change this ring molecule into the pyruvate, NADH, and ATP, like I mentioned. So what happens next, the second pathway, I guess it's really not as much of a pathway as it is one reaction, but pyruvate has to be modified further, and it's turned into a molecule called acetyl coenzyme A. And this is a molecule which can then go into the next pathway, which is the citric acid cycle or the Krebs cycle. They're the same thing, it's just different names for it, don't ask me why. But this transformation, this chemical reaction of altering pyruvate into acetyl coenzyme A is modified by an enzyme complex. So again, there's another step where you require another protein with a specific chemistry in order to perform a certain task inside of a cell. So let's go ahead and look at the citric acid cycle really quickly. Now, the citric acid cycle is an 8-step circular biochemical pathway, and what it does is turns acetyl coenzyme A into 3 molecules of NADH. It creates CO2, 1 molecule of either GTP or ATP, just depending, which GTP is basically a similar molecule to ATP, and 1 molecule of ubiquinone. And this process occurs over and over again. Like I said, it's circular. Things are always going into the pathway and always going back out. But really the important part is that you get production of NADH out of this citric acid cycle. So glucose, pyruvate, acetyl coenzyme A, and then now you've got more NADH. So you've made quite a few of these molecules up to this point. The next step is NADH is then acted upon by what is known as the electron transport chain. I'm sure by now you've probably heard at some point in your education that the mitochondrion is the powerhouse of the cell, whatever they say. It's the one everybody seems to remember. But the reason for that is because of this electron transport chain. It's found on the inner mitochondrial membrane. And what it is it's a series of electron transport complexes that basically perform the task of changing the chemical energy of NADH into ATP, and I'll show you how that happens here. I'm going to first we'll look at the electrons. NADH is split into a hydrogen ion, just H+, and 2 electrons. Now those electrons are captured by these electron transport complexes embedded into this membrane of the mitochondrion. It's passed through several of them, and then what you get is the electrons are recaptured in the formation of water. That's your byproduct. What's more important though is what the hydrogen does. The hydrogen ion, so it's just an H+, there's no electrons on it, has a net positive charge, and that builds up inside of the mitochondrion, which basically pushes the hydrogens out into the intermembrane space. Kind of like how two of the same pole magnets push apart. Positive and negative charges will do the same thing. So this positive charge builds up and it forces hydrogen into this intermembrane space through a proton channel. Once the hydrogens are there, that begins to build up it builds up a positive charge on the inner membrane space this time, and so now the hydrogen basically is being chemically driven back into the membrane where it came from in the first place. But this membrane is not permeable to these hydrogens, so it has to go through a specific channel. And what it does is it goes through a complex which is called ATP synthase. It's really cool. It's a proton pump, and the hydrogen will go into this ATP synthase, and it twists this synthase. It's like a machine, which it's really amazing. It twists and it forces ADP, which is a predecessor to ATP. That's adenine diphosphate. It basically combines an adenine diphosphate with one more phosphate group, and you get adenine triphosphate at the same time that your proton is released and back inside the membrane where it wants to be in the first place. Or not really where it wants to be, but where it's being chemically driven to. So net reaction is that you have this NADH breaks off into 2 electrons and a hydrogen ion. The hydrogen ions are then forced through a pump, which basically builds, mechanically and chemically, it builds ATP atoms, or molecules, excuse me, which the cell can then use for energy any way it pleases. So again, let me just explain one more time why, how does this pertain to evolution? The short answer is that it really doesn't directly, but what I was trying to do again is show you that even a simple task like turning sugar into a more useful form of energy inside the cell is super, super complex, but that it's all just chemistry. It all follows the laws of nature. It's it's positive and negative charges and electrons and free energy and things like that. These are all thermodynamics. These are all things that guide these processes, but there's nothing magical about it. It's all biochemistry. So I was really just trying to get you comfortable with the idea of a biochemical pathway. There's hundreds of these inside the cell, and show you that there's even mundane tasks are very complex in nature, and that it's all driven by chemistry. So that's really all I wanted you to get out of that. What we're going to go ahead and look at next is the more important part, which is protein production. Now we're going to go ahead and look at protein production, which is really the big focus of this video. This is the part that's most important to the argument against the theory of evolution. You have to understand this in order to understand both what the evolutionists say is occurring inside of the cell and what I'm trying to tell you does not happen inside of the cell. In order to understand protein production, first we have to understand DNA, how it's structured, how it works, and how the cell reads that in order to build proteins. So let's go ahead and look at that. DNA is stored in the form of chromosomes. That's what chromosomes are. You've heard that word thrown around a bit, I'm sure. And what that is it's DNA that is tightly wound up in a structure known as chromatin. DNA is basically, it's coiled around these other proteins and it packs it up really small into these chromosomes, and that's basically how the cell can store very large amounts of DNA in a small space. Now when DNA is being read or copied, it has to be unwound and then it takes on its native double helix structure. So I'm going to go ahead and pull up a little picture here for you so you can get an idea of what DNA looks like when it's unwound in its double helix structure. So here's the structure of DNA, and what you have is on the outside you have what is called the sugar phosphate backbone, and that is, you have these ring shaped structures, which is your sugar groups, and then they are bound together through phosphate groups. And that is essentially the backbone of DNA. And then in the middle here you have what are called the base pairs. You have 4 base pairs, and these 4 molecules are basically the 4 letters of the language of DNA. These 4 control everything. So what you have is adenine, thymine, guanine and cytosine. What they do is they hydrogen bond to each other. So there are two of them that bond together, and you always get adenine hydrogen bonded to thymine and guanine hydrogen bonding to cytosine in DNA. And these base pairs inside of DNA are strung together in a specific order, and the order that they're put together in dictates the sequence of amino acids that are put together to build proteins. So the order of these base pairs is very important. It's a very specific thing, and I'll show you in just a minute here how this sequence is basically decoded, and just through another series of chemical reactions, you end up transferring what is literally a language. It's information is translated into building very specific 3D structures with their own unique stereochemistry and their own purpose inside of the cell. There's really two things about DNA that we want to look at. One is how is it copied, and the other is how is it read in order to program for proteins. So the way DNA is copied, like I said, it's in its double helix formation, and there's enzymes called DNA polymerases. And what those do is they bind to the DNA, and I'll bring up a picture here so you can get a look at it. It's a combination of multiple different proteins that work together to accomplish one function. It's really impressive, and it's all driven by chemistry, like I've tried to emphasize here over and over again. But what it does is it'll unzip the DNA, and that's basically breaking apart the hydrogen bonds between these base pairs. So it separates your double-stranded DNA. That's what dsDNA, double-stranded DNA. It separates those apart by breaking up those hydrogen bonds, and then it reads the base pairs one at a time. It basically travels across the length of the DNA and reads those base pairs, and then it makes a copy of it. So you have your original DNA strand, and then it builds up a new copy of it as it traverses along the DNA. Then behind the polymerase, these hydrogen bonds will reform. So the original DNA then returns to its double-stranded, double helix formation. And what you get then is you have your parental strand still intact, and then your new DNA, it ends up being an exact copy of the original strand. There's no new information created here. It's just a copy. And that's basically that's what has to take place in order to copy your chromosomes before cellular division. So that's how it's copied. Now, as for protein production, we're really interested in here. There's a whole series of events that have to take place, and it involves a lot of molecular machinery. Again, it's all driven by chemistry, but it's incredible. You could accurately describe it as molecular machines here and what goes on. First, the DNA has to be read or transformed. This step is called transformation. And the DNA is read and copied into another molecule known as RNA. It's another polymorph. It's another string of base pairs, but it's not double-stranded. It's single-stranded. There's a couple other differences about it, but essentially it copies the information from DNA into messenger RNA. This mRNA, it preserves the information again, and the sequence of these RNA bases is what programs ribosomes in order to build to build proteins out of amino acids in the right order. So what happens there is your mRNA is bound by a ribosome, it's an enzyme, it's a protein complex, and it reads your RNA 3 bases at a time. This is what's called a codon. It is 3 RNA bases. It's kind of like how a word is formed out of different letters. Well, a codon is formed of 3 bases, and these ribosomes read them 3 at a time, and depending on the sequence of those 3 bases, it determines what amino acid basically comes next. And it builds together these the proteins. Proteins are basically amino acids that are strung together. So the ribosome reads the RNA 3 at a time, and the codons program which amino acids are put together in what order in order to build a protein. And we know which codons program for which amino acid. I'll go ahead and pull that up here for you just so you can see. It's really, it's just like a program. You read 3 codons, you get an amino acid, and then the ribosome progresses along the messenger RNA and across the length of it builds up a protein with a specific sequence of amino acids that has its own unique chemistry, its own 3D structure, and its own function inside of the cell. So again, you start with DNA, you transform it into messenger RNA by use of molecular machining. Then that is read by a ribosome 3 bases at a time, known as a codon, which then tells the ribosome, it doesn't tell the ribosome, it chemically drives the ribosome to add another amino acid into the structure, all driven by chemistry. This all happens just passively, driven by thermodynamics again. And that's basically how proteins are built, and that's how how DNA has that much information to it. So the DNA itself, the order of these base pairs determines the order of the RNA bases, which when read 3 at a time determines the order of the amino acids that build up proteins. So it's really, there's a ton of information there, and it's very specific. There's really not room for mistakes, which is why DNA has proofreading functions inside of the cell. More enzymes, more proteins that correct mistakes, chemical malfunctions or mutations that take place inside of the cell. There's actually structures inside of the cell that correct these mistakes, because that information, it's so important that that information is preserved and not altered. So that's all built into the cell to be able to copy DNA, to be able to transform it into RNA, and then translate RNA into protein production. That's just how much information there is to something even as small as DNA copying or protein production. That's not even addressing the purpose of these different proteins and where all that comes from in the first place. I'm going to stop for just a minute here and try to give you guys a better visual of the actual 3D structures of proteins. In order to do that, I'm going to use a program called PyMol. It runs off of the Python compiler, and it's pretty cool. You can download all kinds of protein files and look at their 3D structures. Let me full screen this here for you, and what you're looking at is actually a protein in 3D structure. This is a DNA polymerase. And if you look, actually, it's kind of hard to make it out here. I'll change that in just a second. But in the middle there, this red spiral looking thing, that's your DNA. And then all of this green and blue part on the outside is your actual DNA polymerase enzyme. And if you look, it's composed of all these different little pieces here. Maybe I can zoom in on something. Yeah. So like if you look at these are different amino acids that are all strung together in a specific order, and the order that those are strung together determines both the 3D structure and the chemistry of this enzyme. Now let me change this really quick so you can see the 3D structure better. Where are you at? There you go. So if you look, here we go. This is a much easier way to visualize the 3D structure. What we've got here is the different colors are different subunits, different pieces of this enzyme, and at least several hundred different amino acids strung together in a certain order, like I said. These spiral looking things that look kind of like noodles are called alpha helixes, and you see these arrow looking things, these are called beta sheets, and they're just secondary structures of proteins. In the full 3D shape, that is the tertiary structure, or if this is a multi-subunit enzyme, then it would be a quaternary structure. But what you see here is the outside, this is your 3D structure of your DNA, or excuse me, of your polymerase, and then the DNA here is a lot easier to visualize. Now if you see your double helix, and you can see here's your sugar phosphate backbone, and there's your base pairs that are hydrogen bonded right there. So one of the things that I heard echoed over and over again while I was in school learning this stuff is structure determines function. And so this is the 3D structure, the physical structure, which is determined by the order of the amino acids. But amino acids don't provide just a physical structure. They also have a chemical nature to them. Like, I'm trying to emphasize that this enzyme works because of chemistry. So let me go ahead and show you the, I guess we'll say the chemical structure, and you can get a better idea of how enzymes work and how their 3D structure and their chemical structure are very specific in order to accomplish a certain task, which with these polymerases, like I said, is to read and copy DNA. So here we go. Here's the electrostatics of it. Red represents a negative charge, blue represents a positive charge. And if you look all around the outside of the protein, you've got these charges distributed. But what we have here is called the active site. Any enzyme should have an active site, and that's where the chemistry takes place. And if you notice this blue here, it's all positive. It's a very positively charged active site. And like I said, structure determines function, and chemically it's the same. So why would the active site of a DNA polymerase have a very positively charged active site? Because the sugar phosphate backbone of DNA is negatively charged. So what that does is, if you look, it actually has this little canal here, and the DNA will find its way in here. Just stuff moves around a lot, and eventually the DNA finds its way into this canal. And the negatively charged backbone of the DNA is highly attracted to this positively charged active site in this enzyme. So the two, I mean, it's thermodynamically favorable, and chemically it's favorable. The two want to be together, the positive charge here and the negative charge of the DNA. So the 3D structure allows for DNA strand to come into this polymerase, and then the chemical structure, the amino acids, some of them have a positive charge, some have a negative charge, some of them are neutral, which is these white ones. And this positive charge allows the DNA to stay in the active site. And if you look, there's a little canal, or a little hole, cavity, I guess you could say, and that would be like where your new base pair would, your RNA or your base pair or whatever have you, would usually be filtering in or out of. So just trying to emphasize again, a very specific 3D structure, which is determined by the order of the amino acids, the specific 3D structure physically and chemically, in order for this enzyme to accomplish one specific task. There's really no room for error. If there was a mutation that changed, one aspect, even a couple of amino acids in here, the chemistry would be all out of whack and it would not work. And essentially what that would do is it would kill your organism. This is very specific, and it accomplishes a task using chemistry and physics. There's no magic about it, but that's really this. Hopefully this will give you a better look at the true nature of proteins and enzymes, and how like I was showing you in the energy production, enzymes would perform a chemical reaction. So you would have 20 or 30 of these different ones with different shapes and different chemistry, all programmed in by DNA in order to, perform a certain task. So there you go. Hopefully that's something you've never seen before and you enjoy it.
It's these that trigger the first phase of the process, reading off the information that will be needed to make the protein. The gene is the length of DNA stretching away to the left. Everything's ready to roll. 3, 2, 1. The blue molecule racing along the DNA is reading the gene. It's unzipping the double helix and copying one of the two strands. The yellow chain snaking out of the top is a copy of the genetic message, and it's made of a close chemical cousin of DNA called RNA. The building blocks to make the RNA enter through an intake hole. They are matched to the DNA, letter by letter, to make an exact copy of the A's, C's, G's, and T's of the gene. The only difference is that in the RNA copy, the letter T is replaced with a closely related nucleic acid known as U. You are watching this process, called transcription, in real time. It's happening right now in almost every cell in your body. When the RNA copy is complete, it snakes away from the nucleus and into the outer part of the cell. Then, in a dazzling display of choreography, all the components of another molecular machine lock together around the RNA to form a miniature factory called a ribosome. It translates the genetic information in the RNA into a string of amino acids that will become a protein. Special transfer molecules, the green triangles, bring each amino acid to the ribosome. The amino acids are the small red tips attached to the transfer molecules. There are different transfer molecules for each of the 20 amino acids. They all carry a specific three-letter code that will be read by the machine. Now we come to the heart of the process. Inside the ribosome, the RNA is pulled through like a tape. The code for each amino acid is read off, 3 letters at a time, and matched to 3 corresponding letters on the transfer molecule. When the right transfer molecule plugs in, the amino acid it carries is added to the growing protein chain. Again, you are watching this in real time. And after a few seconds, the assembled protein starts to emerge from the ribosome. Ribosomes can make any kind of protein. It just depends what genetic message you feed in on the RNA. In this case, the end product is hemoglobin.
Now that you understand how the cell is programmed by way of DNA to create specific proteins and how that process takes place, it's worth understanding a little bit more about proteins themselves because they're really, really important inside the cell. They can do a few different things. One of the things they can do is they can serve as a structural protein, which it pretty much is what it sounds like. It serves as a structure inside of the cell. There's different kinds, but for example, like your muscles, it's a structure built out of protein. Also, they can serve as an energy source. This is not a very efficient source of energy. It's more like an emergency source of energy, but that's something proteins can be used for. But the big thing that I want to focus on is proteins functioning as enzymes. I've thrown this word out a few times now. And they, proteins with specific shapes and chemistry to them can be used to build biochemical pathways, like I showed with the energy production, showing how these enzymes are required in order to change a molecule of glucose through like 20 steps or so, each one requiring a different enzyme to chemically modify this glucose into ATP. So you have to have enzymes to do that because the chemistry itself won't take place without some assistance. So there's a ton of information that determines, building these enzymes as chemical machines. The other thing they can do, they can maintain the cellular chemistry. So enzymes act upon other molecules, and that's the molecules they act on are called substrates. And I haven't really covered it, but there's a lot of different substrates that enzymes will alter to perform a specific function, or just to maintain a certain cellular environment, we'll put it that way, a certain chemistry inside of the cell. And that's all done with enzymes. And one type of enzyme is like the DNA polymerase, like I mentioned. So, enzymes program the machine, or enzymes are the machines that copy and read DNA and translate that DNA into proteins. So since you need enzymes in order to copy and read DNA, it kind of becomes the argument of which came first, the chicken or the egg. And this is a really a major hurdle for the evolutionary community to explain. I would love to hear an explanation for this, especially in terms of, if you look far enough back, the origins of life itself. So you have DNA, which I've already tried to emphasize is it's a ton of information, and it's very specific information, and it can't be altered without consequences. This DNA is read by enzymes, read and copied by enzymes, and the information is also translated to build proteins. Well, these proteins that are built by it are sometimes the enzymes that copy and reproduce DNA. So, you know, it's like I said, it's like a computer that writes its own operating system. A computer has to have the programming ahead of time, but if a computer were to write its own operating system, it would have to have the operating system first to write that information out subsequently. And all the machinery has to be there. It all has to be there at the same time. It can't arrive one piece at a time. It has to all be there at the same time. And that's that's just the way the DNA works. That's the way the information is maintained and copied and read. It's self-dependent. I'm going to go ahead and shift into the last topic for this video, which is cellular division. And again, I didn't give you all the details of protein production, but hopefully I gave you enough to really show you just how much detail there is involved in simple processes in the cell, and how we know step by step how proteins are produced. You know, the information that codes them, how that programs ribosomes to build amino acids in a certain sequence, and how those build up to three-dimensional structures with specific chemistry to perform a certain task inside of the cell. That's all I was really trying to show you for that. But we're going to go ahead and move on now to cell division. And what I'm trying to show you here is just how DNA is passed on to future generations inside of, on the cellular level, I guess. And the way this works is under the right chemical conditions inside of the cell, which means that throughout the lifetime of the cell, it will build up these marker proteins. Basically, they signal to the chemical machinery of the cell. They set up the conditions so that cellular division will take place. It's kind of complicated, but basically just the idea that the chemical conditions inside of the cell are correct, and the cell division takes place now. And the first step to that is called prophase. What happens in prophase is, if we're talking about a eukaryotic cell here, which is mostly what we're focused on, is the DNA, which is found in the form of chromosomes in the nucleus, is released from the cell's nucleus into the cytoplasm, where it then unravels into its more native double helix formation. And then that DNA is copied 100%. Usually the DNA inside of a cell is not copied all the way. It's just like a specific gene in the DNA is copied or read or what have you. But in the case of cellular division, it is a total 100% copy. And that takes place in the cytoplasm, and then this DNA reforms into chromosomes. So then you have a copy. So you've actually got, inside of a human cell, you've typically got 23, you would then have 46 once this copy is made. And then the next stage, after the DNA has all been copied and reorganized into chromosomes again, is called metaphase, and these chromosomes are basically lined up at the center of the cell. It makes a straight line right down the middle of the cell, and the pairs are lined up side by side. It's really wild. I don't even know actually how all that works. I can't remember. It's been quite a while. But what happens is during metaphase, it all lines up right down the middle, and then anaphase begins. This is the next step called anaphase, where fibers will actually grow out of the ends of the cells and attach to the chromosomes and pull them apart. So they were lined up as pairs down the middle during metaphase, and then these fibers will actually pull the pairs apart and pull them to opposite ends of the cell. And that's called telophase, when they're on opposite ends. So you've copied DNA, and then you've separated the copies out. And then the last step is for the cell membrane to split right down the middle. And then what you have is the original cell, and you have a new cell with a copy of the DNA. And that's really, I say that's all there is to it, because that's, as far as I'm describing it, that's all that happens, but there's a lot more that goes on. But as far as we're concerned for this, that's really what I'm trying to show you, is the DNA is again, it's another 1-to-1 perfect copy from the parental cell to the offspring. And that's cell division in a nutshell. I'm not going to go into more detail than that right now. Now that's really all that I wanted to cover for this video, and it's already quite a bit, but maybe you're having a little bit of trouble figuring out how this all ties into the argument on evolution. So let me kind of explain what I was really trying to get at here. As per why I showed you the topic of energy production, showing the biochemical pathways required to turn glucose into ATP. The reason I showed you that is so that you can understand that all events inside of a cell are driven by chemistry and physics, the natural laws, I guess you could call them. There's nothing that happens inside of a cell that isn't driven by chemistry and thermodynamics. It's, cells do not have will, they do not have intentions, they don't have motivation, they don't make choices or decisions, they're just chemical machines. And that's really what life is a bunch of chemical machines. No decisions being made whatsoever, if I can emphasize that enough. And that's really why I wanted to show you something like the production of ATP, and also to help you understand how biochemical pathways, kind of what they look like, how they work, and how any single task inside of a cell can require 20 or more chemical reactions to take place just to accomplish one task, and that's all very tightly regulated and highly controlled and highly specific. There's no room for error with something like that. Now for DNA, what I was hoping to do is get you a much better understanding of DNA in general. You understand the structure of it better now, and really hopefully you understand how specific the information is contained inside of DNA. It's not just genetic mush. There's a specific order to it, just like a language or a computer program or what have you. And the amount of information present in DNA is mind-blowing. Like, there's so much information there, and it's all present, and there's not really room for mistakes to be made. That information is that's the whole purpose of DNA, is it's the programming of the cell. And we scientists know very clearly how that information is translated into the structures inside of the cell. Hopefully you got that out of it. And lastly, let's see, I want you to understand that with the cell division, and also with studying DNA, but also studying DNA and cellular division, I want you to understand that DNA information is preserved perfectly, with the exception of mutations, which I'm going to cover in my next video. DNA, all that information has to be kept true to its original sequence, and there's no machining in the cell to change that information. Changes occur as an accident, and they're unguided, they're undirected. There's no mechanism in the cell to change its own DNA. It's always copy. It's never changed. It's always copy. And like I was trying to show you with cellular division, that DNA is copied one for one and then separated out into a new cell. So there's just trying to build an understanding for you before we start addressing really getting into the argument against evolution. Understanding where all this information comes from inside of a cell and how it's preserved. It is not changed. There's no machining inside of a cell to change DNA. It doesn't exist. So the only thing that could cause DNA to change is genetic mutations, which is what the evolutionary community says in the first place. But we're going to go ahead and cover how that could happen in my next video, and hopefully after what you saw in this video you can understand that there's no room for error with DNA. That's really what I'm getting at. There's tons of information. It's copied perfectly. There's no changes made to it throughout subsequent generations of a cell, and there's no room for error. If you get nothing else, that's what you should get out of this video. In the next video, I'm going to be covering genetic mutations, which is where you really start to get into the whole battlefield of, is evolution possible, and how, evolutionists say that it happens and how I say that it doesn't happen. That's going to be for the next video. Hopefully you enjoyed this and you got something out of it, and I will see you next time.
I'm with the